Good morning, everyone. We have this little session to tie up your knowledge about T-cell function. And let's see the next slide, please, Barry. It shows that Dr. Broder, Dr. Jacques, and myself have no commercial relationships. And I'm going to show you an interesting case. This is Michael, who's a 60-year-old man with HIV. He has not been compliant in taking his anti-HIV therapy and now complains of pain when swallowing and a burning sensation in his mouth when eating hot or spicy food. He also has developed destructive changes in his fingernails, and you can see horrible looking fingernails over there. And when they are scraped, the scrapings are sent to my laboratory at Tufts Medical Center. Now, when the physician looks in Michael's mouth, he sees these superficial white patches. You can see the tongue and the mouth. Swabs of these are also sent to the lab, and the report arrives 2 days later indicating that Candida albicans, a fungus, has been isolated both from the mouth ulcers and from the nails. What you're looking at over here is what we call thrush, usually associated with vaginal candidiasis, but you can see these white plaques, which are very typical of Candida infection in the tongue and oral cavity. Now, extensive candidiasis such as this is associated with defective cell-mediated immunity. If you ever see this sort of situation, you're thinking T-cells, cell-mediated immunity, and because we are aware of Michael's HIV status, we measure his T-cell subset levels and the CD4 to 8 ratio in his blood. And from the table, you can see that he has about half the number of CD3 positive cells, that means T cells, but markedly reduced CD4 positive cells and slightly increased CD8 positive cells with a very abnormal CD4 to 8 ratio. And this is what we used to see in HIV patients before there was any therapy. So you know, or perhaps will hear, that HIV binds to the molecule called CD4, and that's how it gets into CD4 cells. And untreated HIV infection eventually results in markedly decreased numbers of CD4 positive cells, such as in this man, and then onto the real immunodeficiency that we call AIDS, which is acquired immunodeficiency syndrome. So Michael's really in trouble here with not taking his treatment. Next one, please. So we are now going to move to breakout rooms for about 6 students to discuss the following questions. Please begin with your introductions, and we will reconvene in about 8 minutes, and each student will be able to answer the questions via poll everywhere. Okay,
all
good.
Right.
Thanks, Barry. So the question here is what markers found on all T cells? And the question is that CD3 is found on all T cells. CD40 on a subpopulation of T cells. And thank you for not saying 4 and 8, and obviously not 19, which is only on B cells.
So just for clarity there, it's so CD40 ligand, the molecule that interacts with CD40, it's CD40 ligand that would be found on T cells.
Yeah. Then they say, which of the following cells would have low counts in Michael's blood? So let's see what you did over here. Yes, so you said Th1 cells and Th2 cells. And that is correct because the HIV virus hits the CD4 molecule, it attaches to it and gets into all cells with CD4 on the surfaces, which are Th1 and Th2. NK cells do not have CD4, B cells don't have CD4, and neutrophils do not have CD4. Would this patient respond normally if immunized with a shingles vaccine? And this is a protein purified from the virus is called herpes zoster, that causes shingles. So if we gave this man a virus, a shingles vaccine, would he respond normally? Peter, would you like to look at this?
Sure. So a patient that is immunized with just a pure protein is not going to make proper antibodies to the shingles vaccine if there's no T cell help. Remember, you need the CD4 help for B cells. So there wouldn't be a normal antibody response or a normal CD4 response in this patient. Now there'd be a little bit of a response. No, basically there'd be not a good response.
Yeah, I think the key word over here is that to make good antibodies, most good antibodies, you need CD4 cells. You need T cell help. That's why we call them T helper cells. And then in this question, would this patient respond normally if immunized with a polysaccharide vaccine for strep pneumonia that generates primarily IgM antibodies? Very good. So the majority of you realize that to make IgM, you do not really need T cells. Pete has told you this a number of times, that B cells can make IgM. They have it on their surface and can make it. But to switch to IgG, A or E, you need T cells. So this is the correct answer, that they would be able to make IgM against this particular antigen.
Yeah, so this is a little bit of a difficult question or a thought question. Just the point to remember is that you should realize that T cells are absolutely blind to anything except proteins. They see peptides. So a T cell receptor simply does not recognize polysaccharide. If you have a polysaccharide vaccine, that means that you can get an immune response to just a simple polysaccharide, but it's a strictly B cell response. And because you don't have T cell help, you won't get class switching or memory cell production in the germinal center. You simply get some plasma cells for IgM. Is that okay?
So now we have a question. HIV infection leads to greatly increased susceptibility to tuberculosis and fungal infections such as Candida. This is a result of a deficiency in which of the following? So this is interesting. The question is, HIV leads to greatly increased susceptibility and fungal infections. This is a result of a deficiency in which of the following? Now, tuberculosis is an intracellular bacteria that requires T cells to kill it. So the T cells will produce factors like interferon gamma that will make the macrophage kill the TB organism which is living inside it. So fungal infections such as Candida require T cells, particularly Th17 cells. So in fact, all the T cells will be defective in HIV infection, including, by the way, Th1, Th2, Th17. But certainly, remember, neutrophils do not get affected or infected with HIV.
Okay, so we're going to go through the case 2 right now, and I'll try to make it fairly quick so we can get into our breakout rooms and have time for you to have discussions. So this is a case where a 4-year-old going to a friend's birthday party a few days later complains of a sore throat, cough, runny nose, and a rash. And the child's name is Dana. And the pediatrician is told that that the dad is actually not sure whether or not Dana has received all the immunizations against childhood diseases. So the pediatrician suspects possibly measles, given the rash, and then confirms his diagnosis on the next slide by the finding of a white spot with a red background inside the child's cheek. So the next slide. And again, this is called a Koplik spot. And this is something that physicians used to look for all the time. And when I was Dana's age, before the vaccine, and people like me actually got wild type measles frequently. This was just diagnostic to be able to, because it actually, even before the rash, you would see these very distinctive white spots on the cheek. So we're constantly looking for these in children that were coming in with, fever and runny nose and so forth. So have the next slide. So on this slide, and we could spend a lot of time on it, we won't, but the take-home message of this shows that after infection with measles, there's a good amount of time while the virus replicates and the adaptive immune response takes off, but has to have some days to form. And you see the first thing that happens in the adaptive immune response is an increase in CD4 T cells. So remember, the measles is growing. The dendritic cells have picked up measles virus proteins, brought them to lymph nodes, and turned on CD4 cells. Following the CD4 cell activation, we see the spike in CD8 cells, which corresponds with the rash, because the rash is actually largely the result of this cell-mediated immune response fighting the virus in the skin. And the CD8 cells go up and go down, the rash disappears, and then later on in the course of the immune response, you have an early IgM response, which is slightly obscured on this chart, followed by a later IgG response as you get class switching. And it's the CD8 positive cells that are clearing the CD4 and CD8 cells that are really clearing the virus from the individual, and then the IgG and IgM are mopping things up and providing long-term immunity. Okay, next slide. Okay, so we're going to go into the breakout rooms. We have just a few questions for you. We've allotted 6 minutes, and then we'll see you. We'll see you back.
All right, Peter. Looks like everyone's back.
Okay.
Hey, look at that. Wow.
Okay, so that's perfect. T cells recognize peptides in MHC, and CD8 cells specifically see MHC class 1 molecules presenting a peptide. So it looks like that's a major point, and it looks like we're good. So we can move along. And this just shows a figure of that, of the CD8 cell recognizing a peptide bound to the groove of peptide binding groove of class 1 MHC. Okay, next slide. Okay, again, fantastic. That the other thing, the second thing we want you to remember is that the killing mechanism of CD8 cytotoxic CD8 positive T cells, which are cytotoxic T cells, kill by inducing apoptosis, and they do it in two ways. One, by punching holes in the membrane and introducing granzymes into the cell to induce apoptosis, and also the induction of apoptosis via the FAS on the infected cell with a molecule called FAS ligand on the CD8 cell. So we have the next slide, just gives you a little diagram of that. So again, FAS is a death receptor, and the FAS ligand appears on these cytotoxic cells, and this interaction generates or induces apoptosis. So neutralizing antibodies would not be involved in killing at all. Okay, next slide, Barry. Thank you. Okay, again, so the major point here is that CD4, the CD8 T cell response also typically requires CD4 T cell or T cell help. And CD4 cells will help activate CD8 positive T cells. They're going to activate measles virus specific B cells. So the helper cells are helping CD8 cells, B cells, and they also do help memory cells. We may not have told you that explicitly, but activating CD8 positive T cell that are memory cells also require the CD4 T cell help. Obviously, well, not obviously, but the activating the alternative complement pathway, which is an innate part of the innate immune system, does not have anything to do with CD4 cells. A patient with HIV/AIDS who has very, very low or no CD4 positive T cells, the alternative complement pathway and the lectin complement pathway in those individuals is fine. Okay, so we're going to, let's move to the next case. These are, yeah, I think we should just move to the next case.
Yep.
So we'll be quick. This is a story of Lisa, 16-year-old, who became very ill with fever, general muscle aches, dizziness, high temperature. She was rushed to the hospital. She lost consciousness, developed a rash over her arms, which spread to most of her body. Very high temperature, very fast heart rate and respiration, and a very low blood pressure of 83/45. She's really sick, okay? She has a bright red rash of flat raised lesions on her trunk and her extremities. She's asked about her last menstrual period had been 6 weeks earlier, and she developed vaginal bleeding on the day previous to this incident when she had inserted a tampon. Given her critical status, extensive lab tests were conducted. Her white blood cell count was raised with the predominance of neutrophils. And cultures of her blood, urine, vaginal fluid, and CSF were performed. And the vaginal culture was positive for abundant Staphylococcus aureus. And Lisa was diagnosed with staphylococcal toxic shock syndrome, resulting from exposure to an antigen produced by staphylococci, which we know is a super antigen. Fortunately, she was given cephalosporin in time with a lot of intravenous fluids, and she improved over the next 2 days. Very, very sick young woman, very lucky to get over this, this whole thing. So we now have a few cases to look at. Do we have time for them, Barry?
We have, how much time do we have left, Peter? This is till 9:25, so we're running short on time. Should we just do these as whole group items?
I think so.
Okay, so we're just gonna skip the small group and scroll into the questions themselves, guys.
So this is a question of how do superantigens activate T cells? Dr. Jacques showed you in one of the diagrams how they work, but let's see what you folks think about this.
Great.
So very good. These superantigens cross-link MHC and the T cell receptor outside of the usual MHC class II binding site. Okay, it's outside. So the first question, they do not bind the groove. The second one, they are degraded peptides and bind to the groove. That's incorrect. And the last one, the superantigen bypasses antigen-presenting cells and directly activates multiple T cell clones is also not correct. The correct answer is it needs both antigen-presenting cells and T cells, but binds to the outside of the groove. Very unusual thing to happen. And there's a beautiful picture of the MHC class II in yellow, the T cell receptor in blue, and the super in white in the middle would be a normal antigen being presented in the usual way. But look at this super antigen coming in from the outside and activating the T cell to produce masses and masses of cytokine, which cause the toxic shock syndrome. And here again, what are the effects of superantigens activating so many cells simultaneously? So perhaps you'd like to spend just a moment on that. Okay, so because of the time, you're right that there will be massive cytokine release by the T cells because they are so activated. Massive production of IL-1 and TNF by monocytes. Now, that will also happen because remember, antigen-presenting cells include monocytes, macrophages, and dendritic cells. Induction of the acute phase response characterized by fever and production of IL-6, and that is also correct because once again, antigen presenting cells are also activated. And the last one, toxic shock leading to low blood pressure and multi-organ failure because of the massive release of cytokines. So in this case, in fact, all of these, remember, both T cells are activated and antigen presenting cells are activated, and all their products will pour out of these cells, giving rise to hypotension and multi-organ failure.
All right, so I think we're at time. Is that true?
Yes, we need to finish. We need to release them. Go on and do things. So thank you. The fourth case, we will have the answers posted on Canvas, but also the three of us are going to actually go through the case and have it recorded. So it'll be when Dr. Jacques edits the recording, you'll have that if you actually want to hear us go through it. Okay, so I guess we say goodbye and thanks. Any questions, drop into office hours or send us an email.
Yes, thanks everybody.
Okay, so let's just go through this case 4. This case is about Michelle, who was had a normal birth and was well, a child, and then when she was almost 2, her mother noticed that she wasn't eating well, had diarrhea, was losing weight. The pediatrician noted that Michelle had enlarged lymph nodes and ordered an ultrasound CT scan for the chest and abdomen. And this showed enlarged lymph nodes in the mesentery and para-aortic region, and she was referred to Tufts Medical Center. So biopsy of the lymph nodes showed marked macrophage proliferation, and they did the lab did an acid-fast stain for mycobacteria, and this showed that there were numerous mycobacteria organisms within the phagocytomes of the macrophages. And the diagnosis was Mycobacterium avium intracellular, and which was cultured from the lymph nodes and the blood of the child. Okay, so moving. And a family history revealed that Michelle had 3 male cousins of a similar age, 2 of whom actually had similar mycobacterial infections, which would not be common. And Michelle and her family members were studied and found to have a defect in the interferon gamma receptor gene. So there's a diagram of it here, and the point here is that the defect in this patient, the genetic problem, is a defect in the receptor for the cytokine interferon gamma. And the point on this slide, for you to remember, the general point, which we've mentioned before, is that cytokines work by binding receptors on cells. So you have to have the cytokine being produced by some cells, and the cells that are activated by that cytokine must express a receptor that the cytokine can bind to and cause signaling within to that cell. So Michelle is missing the ability for interferon gamma to activate interferon gamma receptor bearing cells because she doesn't have the receptor. Next slide. We'll skip the breakout rooms because we don't have any students left. Okay, so the first question is fill in the blank. Mycobacteria are what's actually hiding under there? Mycobacteria are killed when the cells the microbe resides in are activated by blank. In other words, the mycobacteria are living in a cell, and then they will be killed when that cell is actually activated to become a better killer cell. And the answer is okay, it's Th1 cells. No one got it right because you weren't here, but you all hopefully would have gotten it correct. So it's and we'll show in the next slide, the Th1 cells are going to activate macrophages. And the macrophages, you recall from the case, are the cells that are actually harboring the proliferating infection, the mycobacterium. So what's necessary in this mycobacteria infection and also in tuberculosis would be the same, is that macrophages, that the bug has is able to get into the phagolysosomes, the phagosomes of the macrophage and proliferate there. And the macrophage can only effectively kill them if the macrophage is activated by interferon gamma. The next slide, Barry, will show that. Well, actually, it's right. So here you have from your lectures, the Th1 cell makes interferon gamma, which of course is the cytokine that interferon gamma receptors recognize, and a macrophage that has been infected and is harboring the mycobacterium will be able to effectively kill the mycobacterium, but it needs to be activated by the cytokine interferon gamma. Again, that's the cytokine, the signature cytokine of Th1 cells. So if we go to the other question. So given Michelle's defective interferon gamma response, because she doesn't have the receptor for interferon gamma, what other types of pathogens would you expect her to be particularly susceptible to? So the choices are viral infections, fungal infections, extracellular bacterial infections such as streptococcus or staphylococcus, or helminth infections, that is, infections with worms. And the answer is that viral infections and fungal infections would also be a problem for Michelle, because all viral infections require Th1 responses and interferon gamma responses in order to activate cells of the innate system and to fight the virus. And fungal infections are also going to be a problem because of this impaired cellular immune response, cell-mediated immune response. Now remember, extracellular bacterial infections like strep and staph, that is bugs, bacteria that are growing outside of the cells, in the tissue space, but outside of cells. The way we mainly fight those are with complement activation and antibodies. So the humoral immune system and the complement system are the important ones for extracellular bacterial infections. You remember that agamma-globulinemia, people without antibodies will have problems with extracellular bacteria. And as Dr. Rabson explained to you, if someone doesn't have a C3, for example, of the complement system, they have severe recurring extracellular bacterial infections such as staph and strep and so forth. And finally, the worm infections or the helminth infections, the major immune response to those will be a Th2 response, and will be which results in IgE production, and it's the IgE mast cell eosinophil response that is going to be important for immunity to worms. And do we have a?
Just an optional exercise if students are interested. Okay. To complete a table that compares and contrasts the different T cell subsets.
Right. Okay, and we will post the filled out table on Canvas. So, for those of you that watch finish watching this recording, thanks. A great day or evening or whatever it is that you're about to do. Okay.
Thank you, Peter. Barry, thank you.